Hello students, today we are going to study meiosis from the chapter Cell Division. Meiosis. The term meiosis was coined by J.B. Farmer in 1905. It takes place only in reproductive cells during the formation of gametes. Reductional division. The cells in which meiosis takes place are termed as meiocytes. Meiosis produces four haploid daughter cells from a diploid parent cell. Now, on the screen, we can see meiosis as well as mitosis. Meiosis, ये parent cell है. This is parent cell is diploid, and after meiosis, this diploid parent cell divides to form four haploid daughter cells, right? And mitosis में क्या होता है? Diploid parent cell divides to form two diploid daughter cells. अभी हम लोग meiosis start करते हैं. Meiosis is of two types. First meiotic division or heterotypic division. Second meiotic division or homotypic division. The first meiotic division or heterotypic division. Hetero मतलब different. अभी ये different क्या है हम लोग देखते हैं. During first meiotic division, diploid cell is divided into two haploid cells. The daughter cells resulting from this division are different from the parent cell in chromosome number. Hence, this division is called heterotypic division. Meiosis 1 is again divided into karyokinesis 1 and cytokinesis 1. Karyokinesis matlab division of nucleus. So, it consists of phases like prophase 1, metaphase 1, anaphase 1, and telophase 1. Now, about the prophase 1. It is the most complicated and the longest phase of meiotic division. It is further divided into 5 substages. First one is leptotin or leptonema. अभी ये lepto मतलब क्या है? Leptose मतलब thin and nema मतलब thread. So यहाँ पे leptotin में thin thread-like chromosomes are present. So we can see here in this diagram, cell is there. Outermost covering of cell is called it as cell membrane. And inside this, enlarged nucleus is present, right? And inside this nucleus, or this is the wall of the nucleus, which is called it as nuclear membrane. And inside this, के अंदर क्या है? Nucleolus है and chromosomes, which are long, thin, and beaded thread-like structure. Now we will proceed. Zygotin. Zygotin मतलब क्या है? Zygo matlab pair. It is characterized by pairing of homologous chromosomes, right? यहाँ पे homologous chromosomes का pairing होता है. मतलब क्या होता है? देखो, ये homologous chromosomes है और pairing of homologous chromosome उसको synapses called synapses, and paired chromosome bivalents or tetrad. So these two are the homologous chromosomes, and the pairing of these chromosomes are called it as synapses, right? So pairing of homologous chromosome is synapses, and these paired chromosomes are bivalent. दो है इसलिए bivalent, और एक ही क्रोमोसोम के ये दो क्रोमैटिड है, इसके दो और इसके दो, so tetrad. And in between these two homologous chromosome, what is present? Synaptonymal complex. मतलब क्या है? Zipper-like structure assembled between the homologous chromosome, right? Is called it as synaptonymal complex. अभी पैचिटिन और पैचिनिन. पैचेस का मतलब क्या है? थिक. पैचेस का मतलब क्या है? थिक. सो each bivalent or tetrad consists of 4 chromatids. अभी ये देखो 1, 2, 3 and 4. And each pair is united by centromere. In this stage, crossing over occurs between two non-sister chromatids of bivalent. Crossing over होता है यहाँ पे. तो non-sister chromatid means what? See, ये एक क्रोमोसोम के ये 2 क्रोमैटिड है, उनको क्या बोलते हैं? सिस्टर क्रोमैटिड्स. एक ही क्रोमोसोम के 2 क्रोमैटिड्स को सिस्टर क्रोमैटिड्स बोलते हैं. इसके 2 ये सिस्टर क्रोमैटिड. सो one from this chromatid, one chromatid from this chromosome and another chromatid from this chromosome. These two are called it as non-sister chromatids of homologous chromosome. And crossing overs occurs between the non-sister chromatids of homologous chromosome. And due to this exchange of genetic material, or we can say chromatid segment takes place, which results in recombination that leads to variation and thus evolution of the organism. कभी कभी क्रॉसिंग ओवर के ऊपर क्वेश्चन पूछा जाता है. सो यू हैव टू ड्रॉ दिस डायग्राम ऑल्सो. यहाँ पे 2 होमोलोगस क्रोमोसोम है. देखो यहाँ पे क्रॉसिंग ओवर हो रहा है between the non-sister chromatids of homologous chromosome, and when the crossing over takes place, the egg-shaped point of attachment between 2 non-sister chromatids is called it as chiasm, right? And because of this crossing over, recombinant chromatids are formed. अभी डिप्लोटिन. डिप्लोज मतलब क्या है? 2. After crossing over, homologous chromosomes start repelling, दूर हो रहा है, from each other, but remain attached at the chiasmata. डाइकाइनेसिस मतलब यहाँ पे क्या होता है? टर्मिनलाइजेशन. देखो, शिफ्टिंग ऑफ chiasmata towards the end of chromatid takes place. Nuclear membrane and nucleolus completely disorganize and disappear, and spindle fiber starts to appear in the cytoplasm. अभी that is the end of prophase 1. मतलब प्रोफेज 1 में कौन से सब स्टेजेस है? लेप्टोटिन, जाइगोटिन, पैचिटिन, डिप्लोटिन एंड डाइकाइनेसिस. लेप्टोटिन में क्या होता है? क्रोमोसोम स्टार्ट टू कंडेंस. जाइगोटिन में सिनेप्टोनिमल कॉम्प्लेक्स फॉर्म होता है. पैचिटिन में डीएनए एक्सचेंज बाय नॉन सिस्टर क्रोमैटिड्स ऑफ होमोलोगस क्रोमोसोम. डिप्लोटिन में क्या होता है? चाइस्मा विजिबल विद इन बाइवेलेंट. And dikinesis, prophase ends, nuclear membrane disintegrates. Now metaphase 1. An imaginary line, that is equatorial plane, develops at the center of the cell. The pair chromosome, that is bivalent, arrange at the plane in such a way that they lie equidistant, right? Equidistant on either side of plane. Their arms, right, towards the equator, and centromere towards the pole. Spindle fiber formation is completed, which helps in arrangement and movements of chromosomes. Anaphase 1. Homologous chromosomes are pulled away from each other and carried towards the opposite poles by spindle apparatus. Reduction division takes place in this stage. Chromosome number is reduced to half of the total number. Spindle fibers start condensing, become shorter, and pull chromosomes towards the opposite pole. Both sister chromatids of each chromosome are now different in terms of genetic content, देखो, as one of them has undergone recombination. Telophase 1, chromosomes reach the opposite poles, nuclear membrane and nucleolus reappear. Haploid number of chromosome after reaching their respective pole becomes uncoiled and elongated. 2 daughter nuclei are formed. Cytokinesis. It occurs by the formation of the cell plate in plant cell, which is formed at the equator of the dividing cell. Two haploid cells are formed. Second, meiotic division or homotypic division. During this division, two haploid cells formed during first meiotic division divide further into four haploid cells. This division is similar to mitosis. The daughter cells formed in second meiotic division are similar to their parent cells with respect to the chromosome number formed in meiosis 1. Hence, this division is called homotypic division. Meiosis 2 consists of two substages, karyokinesis 2 and cytokinesis 2. Karyokinesis 2 consists of following stages. Prophase 2, nucleus enlarges. You can see nucleus enlarges. Chromosome becomes prominent, shorter and thicker. Nuclear membrane and nucleolus disappear. And spindle fibers are formed between the centrioles. Spindle fibers are formed between the centrioles. Metaphase 2, chromosomes get arranged at the equatorial plane. Some spindle fibers are attached to the centromere. And some are arranged end to end between two opposite centrioles. Anaphase 2, sister chromatids of each chromosome separate and migrate to opposite pole. देखो अभी migrate हो रहे हैं opposite pole में. लेकिन कैसे splitting होता है? You can see here one chromosome. This chromosome splits longitudinally and separates the two chromatids. And then they move towards the or migrate towards the opposite poles. The separated chromatids become daughter chromosome and move to opposite poles due to the contraction of the spindle fibers attached to the centromere. यहाँ पे contraction हो जाता है. Telophase 2, nuclear membrane, ये देखो, and nucleolus reappear. 4 nuclei are formed. Cytokinesis 2, in plant, cell plate formation takes place. At the center of the cell, it forms 4 haploid daughter cells. तो ये देखो, ये parent cell diploid which divides to form 4 haploid daughter cells. That's all. Thank you.